Good morning, everybody. Good morning to folks online. We're going to go ahead and get started. For those of you who are in the room, thank you again for showing up. And for those online, we're going to do the same thing that we did yesterday. If you're relatively new to the online system, we're going to be using a system called Slido, SLI.do. And the Slido event code is in the bottom right-hand side of the event slide. So if you log into SLI.do and sign in with PSD, I'll make the joke again, it's not Pumpkin Spice Day 19, PSD 19, then we'll use that as our way of interacting with the online audience. And we'll try as the day goes on to address those questions when we come to the point where we're doing questions. So at this point, I'm going to go ahead and turn things over to one of our organizing committee members, Breeze Riley, to get things going.
Good morning, everyone. It's my pleasure to introduce today's keynote speaker. Janet Freeman-Daily is a retired aerospace systems engineer trained at MIT and Caltech, who's also a freelance writer and lung cancer patient and activist. Diagnosed in May 2011, she learned about biomarker testing and clinical trials from other patients in online communities. After her tumor tested positive for ROS1 gene fusion, she has had no evidence of cancer for over 6 years thanks to research. Her focus is now on translating the experience and science of lung cancer for others through writing, public speaking, and research advocacy. Welcome, Janet.
Are you awake? There we go. Thank you to the organizers for inviting me to speak today. I'm going to talk about my experience wayfinding through personal genomic data sharing. It's a meandering route with a few clear signs. But it's a good story of why sharing genomic data for research is worthwhile. What is wayfinding, you ask? Good question. Wayfinding is the process humans and animals use to get from one place to another. Unlike navigating, wayfinding doesn't assume that you can see or know where you're going or what the path is. It's an art practiced by the ancient Polynesians as they discovered new islands in the South Pacific, in canoes much like the one pictured here. The process of wayfinding includes orientation, which is the attempt to determine where you are, route decision, selecting how you're going to go, route monitoring, checking to make sure you're on that route, and destination recognition to figure out if you've arrived at the right place. So whenever you see this canoe on a sign in my talk, I'm talking about wayfinding. So now I'm going to take you along my timeline through personal genomic data sharing. 1990s, Human Genome Project. This geek was really excited that we were going to figure out exactly all the genes and processes necessary to build a human. And it didn't work out quite that way. We know where all the genes are on the genome, but as I learned, we don't always know what they do. So as a conscientious geek, I wanted to make good use of this information. I'm going to do a little wayfinding. Where am I? Well, as far as I can tell, I'm in this big uncertain sea of data. Where do I want to go? Well, I can envision 3 different types of destinations. One is this lovely little tropical island where I could altruistically donate my data, donate my information, and feel good that I improved research. The next was going to a series of interconnected islands on an atoll to find out how me or my family members might be at risk for various conditions. And the third is this rocky little island of dealing with an actual disease that I or a family member might have and trying to find a cure. And the motivation for participating goes up as you go with each island. And I think most patients experience this when they're trying to make a decision about participating. Why did I care about donating? Well, there are risk factors in my family for some major diseases. I have Alzheimer's and dementia on both sides going back at least 2 generations. I have family members, not the extended family, generations back with various learning differences like dyslexia, ADHD, and Asperger's or autism, including my adopted son, who happens to be a cousin. And we have glioblastoma running in the family. My son's birth mother died of it. Her oldest brother died of it. My grandfather died of it. And one of my grandfather's brothers had a brain tumor, which probably was glioblastoma. So I'm really motivated to learn about this stuff. However, in my retirement job, when I started researching all of this, I discovered that it ain't so easy. I was fighting the current, essentially. The only known genetic risk factor for late-onset Alzheimer's is the APOE4 allele, and it's not a strong linkage. The only information about autism is a bunch of genes that might be related, but they're not really causal, so we didn't know much back in 2009. And for glioblastoma, the genetic counselor told us, well, we know absolutely nothing about a familiar linkage, so there wasn't much I could do. I might have a risk for these things, and I might not. So I decided it really wasn't worth the risk for me to try and find out if I had, say, an APOE4 allele, because this was back before they'd removed the pre-existing conditions from insurance, and I could risk losing my health insurance if somebody found that information in my medical file. So my wayfinding, I came to the conclusion that at this point, a lot of the genetic causes are poorly understood. There's no prevention for any of these, and pursuing my data could cause a problem. So I let it go. And then I got lung cancer. The light cough that I had was diagnosed as advanced lung cancer in May of 2011. This was quite a surprise because I had no risk factors and I had a healthy lifestyle. Oh, a little overweight, but we'll ignore that part. At that point, individual molecular testing was just getting started for lung cancer. So my doctor ran some individual molecular tests for oncogenic drivers. So in lung cancer and all cancers, an oncogenic driver is not an inherited risk factor. These are alterations in a gene that you acquired sometime after birth, and they cause the cell to think that it should act like cancer. The altered gene creates an altered protein that's always stuck in the on position, and it tells the cell to invade your neighbors, live forever, make lots of copies of yourself, and go out and invade the neighborhood. So there are new drugs called targeted therapies that can treat these oncogenic-driven cancers. They go in and bind to that altered protein and turn it off, so the cell stops acting like cancer and it dies at its regularly programmed time. At the time I was diagnosed, there were 2 tests available, one for point mutations in the EGFR gene and one for fusions in a gene called ALK. EGFR is important in a lot of cellular processes. ALK, the best we can tell, has something to do with development of a fetus, but it doesn't seem to be active in adults. Unfortunately, I didn't have any of these. So I went through curative treatment, chemo and radiation, and as soon as I stopped chemo, I progressed again. Fortunately, I had been in online groups, and I had learned about genomic testing, and I'd learned about clinical trials, and I found a clinical trial called the Lung Cancer Mutation Consortium protocol, where they would test your tissue for a panel of 10 different genes and several alterations in them to try and find an oncogenic driver. There was only one location that was still open. This was right at the end of the trial, and it was at University of Colorado. And since I wasn't allowed to fly, they graciously allowed me to ship them my tissue and get tested. Unfortunately, I didn't have any of those oncogenic drivers either. So I had more chemo followed by more radiation. Shortly after that, one of the companies that makes one of the chemo drugs that I took decided to do a research study using 23andMe. This is before the FDA shut it down, so they were still releasing genetic data. I looked through it and was interested to find that I had a slightly elevated risk for lung cancer. And then I looked at why. They found that they really didn't know what that gene did, but it was on the genome near to a gene that caused nicotine receptors to be made. So they figured, it makes you more likely to smoke. Really? I never smoked. Don't get me started talking about lung cancer stigma. But I also found that I had a slightly elevated risk for late-onset Alzheimer's. I had one of those APOE4 alleles. Now, that by itself was a little concerning, but not too surprising. What was cool was that when I was in a study for chemobrain at the University of Washington, they discovered that people with APOE4 alleles are more likely to get chemobrain after chemo. So that was kind of cool. I wanted to donate my data to the researcher, and they said, I don't know what to do with this report. So I let that go. However, fortunately, there were other very engaged patients online, and I learned from one patient about a clinical trial for patients with a new gene fusion called ROS1. It was more likely to happen in people who were young, adenocarcinoma, never smokers, and were negative for the other known mutations. So I contacted my doctor and said, let's get this tested. And he said, I don't know how to do that. Turns out the only place in the world that was testing it was at Massachusetts General Hospital, and you had to be a patient there. I was in Seattle. That wasn't going to happen. However, through a series of unlikely events, which if you want to read all of them, you can go on my blog sometimes, I ended up actually getting diagnosed with ROS1 at the University of Colorado, and they had a clinical trial. So I went on this targeted therapy clinical trial in November of 2012. I take a pill twice a day, and after immediately progressing after 2 different rounds of treatment, I have had no evidence of disease for over 6 years. I'm still there. So I'm a big believer in genomic medicine. So while most of yesterday was talking about genetic risk factors, I'm focusing entirely on genomic drivers of cancer. Next step, later in the next year after I started on this trial, I talked to my friend, Jay Lake. This is Jay. Jay is an award-winning science fiction writer who lives his life out loud. He also had metastatic colon cancer and had run out of treatment options. So his friends, many of whom were either medical researchers or cancer patient advocates, decided to create a GoFundMe fundraiser for him called Sequence a Science Fiction Writer. And they raised enough money for Jay to do whole genome sequencing and whole exome sequencing of both his cancer tumor and his healthy genome and have it analyzed. At that time, it was over $40,000. Unfortunately, what they handed Jay was 2 hard drives and said, here's your data. So part of that money went to analyzing it to find out if any of that data would help Jay. Unfortunately, it didn't suggest any clinical treatments for him. However, Jay was not to be deterred, and he decided to donate those hard drives to the Harvard Personal Genome Project. He then also entered a clinical trial here at NIH with Dr. Rosenberg on working on immunotherapy, and they took all of that data on those hard drives to try and figure out how to customize immunotherapy to help him. They said they learned a lot from his information. So, unfortunately, Jay died from complications of that therapy, and yet he came up again the next year. I was at Stanford Medicine X in 2014, and who should appear up on the main stage but Jay. A researcher had accessed Jay's open-sourced whole genome to look at how a certain antibiotic might actually interact with a missing stop codon in a gene in Jay's profile to find out if that could have had something to do with his cancer. All they mentioned of the person is that this was donated by a science fiction writer, and I knew who it was, so you can de-identify people from genomic data. I was really pleased to find out that Jay's desires for making a difference happened because he could donate his genomic data. Also that year, I gave a keynote at the Cancer Center in San Diego, and I discovered in my research the LifeRAF group. There's a condition called GIST, which is a GI tumor, that was the first treated by targeted therapies by a drug called Gleevec. And the LifeRAF group had all found each other in a listserv on the ACOR site and had formed a group where they were collecting data on the experience of living on this drug, and by this point had already started on collecting tumor tissue and driving their own clinical research. This was my first experience with people who had a particular genomically driven disease that were actually researching it. So the next year, I decided I wanted to get more involved in research. I was still going out every 8 weeks to the University of Colorado for my clinical trial, so I asked them if they needed a patient advocate on their NCI SPORE. For those who don't know, the SPOREs are specialized programs of research excellence, and the NCI sponsors them in several cancers. They happen to have one in lung cancer. In fact, it was the oldest one in lung cancer at the University of Colorado. I'm sitting in my very first external advisory board listening to the head of the SPORE repeatedly complain, one, we don't have enough tissue to run some of these studies because lung cancer is usually sampled with fine needle aspirate and there usually isn't enough tissue for testing. And he also kept talking about how patients were dying. He said it several times. I was sitting right next to him and he kept getting louder. It's like, thank you. I know patients die. And then it occurred to me that there were people in this online lung cancer community, family members, who had lost a loved one to lung cancer, who kept saying, I wish I could do something. And I went online during this meeting and sent out a rather delicate query. If you could make a difference in lung cancer research by donating your deceased loved one's tissue, would you do it? Within an hour, I had 75 people saying absolutely, which kind of stunned me. So I brought that up in the meeting, and the project on the screen was born, where the University of Colorado Biobank had a remote donation possibility where people could send their loved ones' archived lung cancer tissue in order to build up the biobank. Well, it sounded like a good idea. This was my education in research. This idea came up in February. We finally had an approved protocol, and this is for non-human patients because they're all dead, in September. And by then, a lot of the family members traumatized by their loved one's death had left the group. We still managed to get a large number of patients donating their tissue. And I liked the idea that people could participate remotely in collecting this data. However, I was a little surprised to find out that if we donated tissue to this biobank, we no longer had any say on what happened to it. I had talked with them about, well, if I donate my ROS1 tissue to you guys, can you say it's only going to be used for ROS1 research? And the answer was no. Well, that didn't feel too good. I wanted to have control of this rare tissue. I don't want what little ROS1 tissue there was in the world to be used for researching diabetes. So I still had some more to learn. Now we're going to do a little route monitoring. Genomic biomarker testing in lung cancer is a big deal, and so are targeted therapies, but they're evolving very rapidly. And as often popped up online, tissue is the issue. There just isn't that much lung cancer tissue, so how do you get it, and how do you ensure it goes where you want it to go? I also realized that both healthcare and researchers don't easily support patient tissue donation. I wanted to donate the tissue from my deceased cousin who had glioblastoma to the biobank because glioblastoma was the first cancer in which they discovered ROS1. I thought, isn't that interesting that my cousins are dying of glioblastoma and I got lung cancer? I wonder if there's a connection. Well, I contacted the hospital. That was my second cousin, and I wasn't closely related enough for them to release the tissue. And the family members who weren't focused on research weren't willing to go through all of the paperwork in order to make it happen. So that tissue never went anywhere. So fast forward, 2015, 5 patients who all had ROS1 positive lung cancer, who were all on the same drug as me, crizotinib, happened to meet up at the Longevity Hope Summit here in Washington, D.C. We all had kids at home, and we all had no next treatment option. And we decided we wanted to do something about that. So we formed a group that eventually became known as the ROS1ders. Isn't that cute? And our mission was to change outcomes for ROS1 cancer patients, but not just lung cancer. There's a lot of cancers that have ROS1. We wanted to do support for patients and caregivers, so like many rare cancers, we formed a Facebook group. And the reason we chose Facebook was because it gave us control of moderating our group and what discussions could happen. I contacted a couple of other formats, and they said, well, no, you can't have access to the names of these people. You can't contact them directly. You'll have to go through it indirectly. It's like, if we're trying to talk to each other about how are we going to donate data and say, oh, yeah, we've got a person on the same drug as you in London, you need to have that information, and Facebook let us do that. But we also wanted to increase awareness and education of our disease because there's maybe 4 or 5 experts in the world who know anything about ROS1 cancer. Most doctors will never see a ROS1 cancer patient, and most doctors don't even test for it. So we also wanted to accelerate and fund research, and we wanted to improve access to both diagnosis and treatment. This is an issue. ROS1, as I mentioned, is rare. It occurs in, depending on who you ask, 1% to 3% of lung cancers. And you can see lung cancer, this disc is all lung cancer, adenocarcinoma patients. It's being increasingly divided up into these genomic drivers. And it also occurs in up to 12 other cancers that we found it so far, again, in about 1%. And ROS1 is a fusion. It's not a point mutation in one gene. It's actually swapping exons between 2 genes, and there's at least 20 fusion variants. And we only had 1 model for all researchers who were trying to study it or trying to make drug development. So we decided we needed to do something about that. We wanted to collect data and specimens to form a biorepository. We also wanted to raise funds because there weren't that many people who had money to research a rare cancer like ROS1. And we wanted to make whatever research products we generated widely available to attract more research. Well, so we thought we were doing well. We had grand goals for our biorepository. We were going to involve patients. We were going to have this open registry. We were going to have it both IRB approved and be able to access the data so we could still connect patients who had similar diseases in similar locations. We were going to link the surveys. We were going to link the genomic results. And we were going to have patients involved in every step. Nope. That didn't happen. In part because we discovered that some of the people didn't have the same vision of partnership that we did, and also they chose an IRB and a contract arrangement with a researcher that said the researcher got to keep the data. That data still has not been really released. If we want to get data for the 100-plus patients who donated their treatment sequence, we have to do it all over again. So there are lessons to be learned along here. Are we heading in the right direction? Well, I think it's important to know if there's mutual trust and respect in the partnership, if there's possibilities of the data being siloed. Will patients find out the results of the research they're participating in? And maybe we need to go a different way. We ended up doing the ROS1 Cancer Model Project. Patients who have an upcoming medically necessary biopsy donate fresh tissue that gets shipped within 24 hours And they can either go with the arm that is creating mouse models in partnership with Champions Oncology or cell line models at the University of Colorado, who created a protocol in response to the biobanking project that I tried to start. And so far, we've had over 5 attempts at PDX mouse models, and there are 3 currently in development. And we've created 4 cell lines that have been genomically sequenced and are available to any researcher who wants to access them for the cost of shipping. The way they fund that is by charging pharma in order to keep the cost of the research going. And we have that information has been used in 3 publications so far looking at resistance for ROS1. Our group, however, is a global community. We have people in over 28 countries. Since 2015, this rare cancer now has over 500 people in the group. And we have 8 different cancers represented in the group. There are also active groups, since not everybody speaks English, in Germany, the Netherlands, China, and Japan speaking their own language, and we have members who translate questions and answers between the groups. However, we can only do these biobanking projects in the U.S. because the cell lines can't grow if something's taking too long to ship, and you have to keep it fresh in order to make the transplantation for the PDX mice work. So some of these other groups, there's a group in the Netherlands that have raised money and are going to be starting their own cell line work, and Australia is looking at doing it also. So what we use this information for is helping patients navigate both treatments and trials. You can see a list of all the treatment options right now that ROS1 currently has. A lot of these are off-label or experimental. Fortunately, most of the drugs that work for the ALK fusion have some effectiveness for ROS1, but not all. One of our big challenges is that a lot of people think they all do, and they'll say, oh, well, alectinib is the first-line drug for ALK, so we'll use it in ROS1, except alectinib doesn't work for us. So one of our big challenges is educating patients and their providers about what drugs work. And another challenge is resistance mutations. Unfortunately, on targeted therapies, everybody eventually develops resistance to the drug. And most patients in EGFR develop one particular mutation called T790M for which there are treatments. Well, eventually, we found out in ROS1, a large number of us have no mutations on the protein. So targeted therapies aren't going to make a difference. And we're still trying to figure out how that works. But I'm particularly proud that this study that shows the resistance mutations used our cell lines. Patients donating can make a difference. There are other groups who saw what the ROS1ders are doing and started their own work. So now there are 2 groups in ALK-positive lung cancer, a group called the EGFR resistors that goes across all cancer types, but primarily focused on resistance in lung cancer. The exon 20 group, which is for very rare mutations that are insertions of HER2 or EGFR, and renegades, which is just getting going. The reason these groups exist is because targeted therapies worked for enough patients that we were alive and feeling healthy enough that we had the energy to want to make a difference. We all are taking a slightly different approach. ALK Positive raised over $600,000 for ALK research. Exon 20 group helps patients navigate between clinical trials because they don't have an approved drug. EGFR resistors, who have several drugs available, now have a drug that causes them to develop other mutations for which there are no treatments, so they're looking at spearheading research into that. And all of these groups are available online. So you've heard a lot of talk about the problem with Facebook privacy. The problem is those groups who want to form spontaneously at the moment don't have anywhere else to go, so we're still there. So here are some of the signs that we found, and this is between talking with Corey Painter for the Count Me In project and our own experiences, we kind of came up with this list of some of the things that are important for patient-partnered research. You need to include all patients from the start. You have to ask questions that are meaningful to those patients. You have to develop patient-centered measurements, accommodate the patient's clinical realities. For instance, when a study said that we needed a washout period of 10 days between TKIs, we all said, excuse me, you do know we have disease flare at 7 days, right? We leverage social media and patient groups. We share progress with the participants, and we make results rapidly and freely available. I would love to say that all of us do this in all of our groups, but most of us have pieces of it here and there, and we're still working on getting the whole picture put together. One of them that's working on doing this is the Rare Cancer Initiative, which is a partnership between the Broad and the Rare Cancer Disease Foundation. I'm not sure I got it right, but they are on the website pattern.org. And like the ROS1 Cancer Model Project, patients can contact them and donate their rare tissue, which is then sequenced at the Broad, and they make cell lines and then they sequence it. They're also trying to make organoids, which are more three-dimensional models than cell lines. They've had 277 patients participate. They've gotten 228 samples. They've got greater than 30 cell cultures, 5 cell models, and have generated new research. So there's hope. There's ways that this patient-partnered research can work. Another one that's in development is the NCI Cancer Moonshot Biobank, of which I'm one of the patient participants helping to design it. And they're trying to do everything right. They're focusing on community sites because 85% of patients get treated in community locations. They're doing not only tumor biopsies, but they're trying to do longitudinal biopsies so that they can biopsy the patient at progression and try and find out how some of these rare diseases change. They're also matching it with blood samples. They want to get diversity across the United States. They have patient and provider engagement. We're helping them to design the website. They are going to return CLIA molecular characterization. So when a patient participates, they get the results of their genomic sequencing. And they're going to try and make some cancer models out of these. This is still in development, but it's a wonderful initiative, and I'm really excited to see where it's going to go. Also, in 2018, a new development happened. For the first time, there was a broad genomic panel that was commercially available for all of the genomic drivers for lung cancer for which there were targeted therapies. And it's also available in other solid tumors. So this is a first. Patients are able to choose whether they want to have their genomic sequencing done or whether they want to participate in one of these studies that will do genomic sequencing. And that raises all sorts of questions. If nothing else, I now have the choice of pattern.org, the ROS1 Cancer Model Project, or the eventual NCI Moonshot Biobank database, where I can donate my tissue. And that's only a little itty bit of tissue. How do I decide which of these options is going to give me the best bang for my buck? So we have some wayfinding needs. These are all questions that patients have when they want to participate in research. And we need to find ways to help them come up with the best ways to do things. We need education. We need changing in our regulatory models so that patients can have autonomy in making these choices. And we need to find better ways to include diversity. I think patient-partnered research can make some huge changes, and I hope you'll consider it in your work. Thank you.